All right, hello everyone. So today we're gonna finish where we left off from lab 1, and we're gonna start lab 2. Okay. Uh, Hopefully all of you are doing well. So whoever did not show up to the optional Zoom meeting last Thursday, I shared the recording with you. It's in the announcements, or if you go to the modules, it's in the course overview. The hybrid one took about 57 minutes. It's worth watching it. You'll watch it one time, but you'll know everything that you need to know for the course, okay? And then if you're interested specifically on the materials on Access Pearson, I would say watch towards the last 10 minutes or last 15 minutes. Okay, if you don't want to watch the whole thing, at least watch that to know all the resources on Access Pearson. Okay, does anybody have any questions? All right, and then whoever just walked in, you can pick up handouts from my desk. And like I said, we're gonna finish lab 1 first, okay? All right, everyone, last week we started lab 1, and then it was a busy lab because we had introduction and we started lab 1. This is where we left off. So we were gonna start talking about homeostasis and equilibrium, right? So our bodies, everyone, is in a condition where it's always trying to reach equilibrium or homeostasis. What we mean by that is, let's say something is off and then it's not normal, then it goes back and tries to do everything it can so that everything goes back to normal. Okay, and then I'm going to give you an example. This is not an example on the human body, and this is with a thermometer and a thermostat. Okay, let's say, and then these days we're using the heater, right? Let's say we've set up the temperature to 70 Fahrenheit, and then the temperature in the room gets cold. Okay, when the temperature in the room drops, right, drops less than 70, then our thermometer in the thermostat is going to detect that the temperature in the room dropped, even though it's set at 70, right? So because it dropped, but it should be at 70, right, the heater starts working again, right? And then it warms up the room until it goes back up to 70. Once the room warms up, it's back to 70, then the heater stops working, right? This is how our the thermometer, the thermostat works. But if you look at it here, we have something called the stimulus. So there should be some signal that something went off. Like, for example, the thermostat detected that the temperature dropped below 70, right? So for us, for example, for humans, again, there should be some signal, something telling our body that something is off, something or is not where it's supposed to be, right? And then we're supposed to reach homeostasis. So we have to make changes, right? So the receptor in this case is the thermometer. It detects the change, and then well, it brings in the message to the control center. The control center for us, it would be the brain, right? And then the brain would check the message and understand the message and interpret the message in the control center. And then this is when we understand that, oh, the temperature went below the point. And then this is where the brain, the control center, has to give a response, right? Which is to turn on the heater. Okay, and then it turns on the heater, and that's the response, right? And until it goes back to normal. Once it goes back to normal, then the heater turns off because the room is back to normal. Okay, this is not a human body example, but this is the example for a thermostat. And then this is the example how our bodies tries to reach equilibrium. If something is off, some signal travels to the brain, and then the signal arrives to the brain, we won't be able to understand what's happening. Once it goes to the brain, the signal goes to the brain interprets the message, understands what's going on, and initiates an appropriate response. And then it initiates the response, that response happens, and then after that, everything should go back to normal. Okay, so let's do one example with the human body, everyone. So we get a stimulus, okay, that the body temperature decreased below normal range. So this is not the thermostat, not the room, us, like we're cold, right? The human, as a human being, let's say we're cold because the room got cold and then we're feeling cold, right? And then our receptors, right? Our receptors in our skin. So we're gonna learn when we learn about the integumentary system, we have receptors, sensory receptors in our skin that are gonna detect the message that, okay, this person is feeling cold, right? And then take the message to the brain. But these receptors, everyone, they're just messengers. They don't know what signal they're bringing into the brain. They just bring in the message. And then once the message arrives to the brain interprets that message, and then we understand what's happening, okay? For example, if you put your hand on a hot surface and then you immediately, right, you take it off so that you don't burn your hand, what's happening is that when you put it there, some change is happening, right? There's a trigger or some stimulus, and then our sensory receptors, they take that message to the brain, and then the brain understands that, okay, you're putting your hand on a hot surface. If you keep it there, you're gonna burn your skin, and then initiates an appropriate response, which is you have to remove your hand, right? But all of this happens, it's a reflex, it happens less than a few seconds, right? But what I'm trying to say is when the receptor takes the message to the brain, it's just taking it, it doesn't know what the message is, right? The brain is the one who interprets the message. And after interprets the message, then we understand what's happening, and then the brain initiates an appropriate response to the effector muscles. So here, the control center is the brain. So the brain cells, they process the stimulus and they determine the body temperature that, oh, this is what's happening. The person is cold, the body temperature went down, and then let's initiate a response to the effector muscles in this case. So basically the nerve cells, they activate our skeletal muscles to start shivering. So you've noticed, right, whenever you're feeling cold, your body starts like moving, right? Your muscles, it starts shivering. Oh, there you go. Starts shivering, and then you produce heat. And then when you do that, your body temperature goes back to normal, and then you stop shivering. Okay, this is the same example of the thermostat, but the actual in the human body, okay? There's another example, everyone. This is the control of blood clotting by a positive feedback mechanism. So homeostasis takes place with either positive feedback mechanism or negative feedback mechanism. It means that if something off balance or if something is off, right? And the body's always trying to reach homeostasis where everything is normal, okay, where it's supposed to be, right? It initiates either positive feedback mechanism or negative feedback mechanism. For example, if we don't have enough of something and we need it, right? For example, if we eat a lot of sugar and then it's gonna produce a lot of insulin so that we can break down the sugar, right? But if we're starving, there's no sugar to cut down in our body to break it down. Is it gonna produce insulin? No, it's not, right? So whenever we don't need it, it needs to stop producing it. And then whenever it's needed, then we need that positive mechanism to keep making more, and making more, okay? This example here, we're gonna say it's an example of a positive feedback mechanism. So everyone, in this example, we get a message to the brain that the person fell down, got injured. And then, so if we do have an injury where it hits or a cut, sorry, let's say a cut, not like that. When you have an injury that hits the second layer of the skin, that's where you have your blood vessels, right? And then you start bleeding, okay? If it's just a scratch on the skin, you don't bleed, but if it's a lower layer, then it means that a blood vessel has been damaged, right? And then you start bleeding. So if you're bleeding, then the blood vessel has damage, and then we need to clot that blood vessel so that it stops bleeding, correct? So whenever we're bleeding, the bleeding is the stimulus, and then the receptors. So there's the receptors on our platelets in our blood that they detect this change that, okay, something is happening, right? There's a damage, and then we need to fix it, right? But the receptors, so this message has to go to the control center, the brain first. And then whenever the brain finds out and gets the message, integrates the message, it understands the message. that, okay, we have a blood vessel that's, injured, and then we need to clot the blood vessel, right? Then it initiates a response, and then the response for the brain in this case would be to activate more platelets because the platelets are the ones that are gonna clot the blood vessel, right? Whatever is it's cut. So basically here we have positive feedback where more and more and more platelets are activated, right? And then activated platelets in the blood, and then the platelets, the response is they seal the blood vessel. Now that the blood vessel is sealed, there's no more bleeding. Then we stop asking the platelets to get activated and clot the blood vessel. Okay, so this is an example of a positive feedback mechanism. Anyone, any questions about this? And then as we go, this is more physiology, but as we go, we explain different things happening in the body throughout the semester. You'll see how, especially when we talk about hormones and glands, how this mechanism takes place, okay, how it happens. Everyone, this slide is, remember, this is part of Lab 1, and then the first week I kept saying about how it's important for us to understand the structure, how things are made up of, right? Because the structure defines the function, right? Things are made in a certain way because of the function that they serve, because of the purpose that they serve, okay? If their structure is wrong, it's not right, it's not appropriate to the function, then it's not going to be able to do the function. This is just an example here. Everyone, you can see here in this image magnified lungs, okay? And then the tissues where, the tissues between the lungs and the blood vessels. All of you know this, I'm not gonna go into details. The blood from the heart goes to the lungs so that it can dump the deoxygenated blood and collect new oxygenated blood and comes back to the heart so that it can pump to the rest of the body, right? So that the blood vessel, the blood has to do exchange where it drops off the carbon dioxide, picks up more oxygen, right, from the lungs, right, so that it can go back to the heart. So there's gas exchange happening here. Okay, and then what we're seeing here in this image, this is the lung, and then this is the tissues that make the lung, and then these are the blood vessels. And then here you can see that these tissues in the lungs, they're very thin. They're simple squamous epithelium. It means it's only one layer, the flat cells, right? It's not thick, which makes it very easy because it's thin layer. It makes it very easy for gas exchange, right? We can exchange oxygen, carbon dioxide to the blood and from the lungs. But imagine if this tissue was made up of many layers, stratified, a thick layer, right? And then you can see from the image, it's trying to show us how, like, the gas exchange, like, it's not gonna happen, or it's not gonna reach all the way because of that thick barrier, right? And then this is just an example that, if the structure is wrong, then the organ is not going to be able to do its function, okay? So we're going to see we have different types of cells, a lot of cells, and then different types of tissues, and then our organs, different organs are made up of different types of tissues because of the purpose that they serve, because of their function, okay? All right, next thing that I wanted to cover was the metric system. Everyone, I believe in your Lab 1 handout, you do have questions about the metric system, but on the exam in anatomy, I don't ask anything about the metric system. And then I'm going to say I'm 99% sure there's no questions about this, but I'm going to go back and check. I'm not saying 100% because now I'm doubting myself. So, but the reason I cover the metric system is because next semester in physiology, not only you have to be familiar with the metric system, you're going to be completing some problems where you have to know the metric system, and then you can have to make conversions. For example, if you have to administer a drug to a patient and the patient is this many, the weight, you need to know the weight of the patient to know how much to give and all that stuff, right? So in anatomy, I would like to introduce it again. I'm pretty sure you've been exposed to this in Bio 5, but we'll go over it again, and so that when you see it again in physiology, you're already familiar, okay? So, so the reason why we're covering the metric system is because here in the United States, in our everyday lives, we do not use the metric system, okay? And then we're the only ones that don't use the metric system all around the world, and especially in the medical field, the metric system is what's used, okay? And so, and then in our medical field here in the US, we heavily rely on the metric system, okay, especially in our fields, okay, for several reasons. And then I'm gonna mention some of those reasons soon. But before we go to the next slide, and I can show you the table, I want to ask you these questions. So some of the metric units you're all familiar with, right? So tell me if you know the answer. For example, when I'm talking about mass, when I say mass, what unit comes to mind? What metric units? How do you measure mass? Sorry. How do you measure mass? And then what's the unit for mass in the metric system? Grams, right? That's correct. Good. And then when I talk about volume, what comes to mind? Liters. Good, good, good. And then what's the unit in metric system for length when I'm talking about length? Meters, okay? So that's good. So those are the basic units that we're gonna talk about in the metric system. And we'll talk about why do we use those bases, okay? So everyone, you just mentioned here that, so we have grams for mass, right? We have liter for volume, and then we said we have meters for length and distance, right? So this is the standard system of measurement, and then these are the base units that you're seeing in the picture. Don't worry about the time, the second, because it's the same. And for most of these, it's usually the reason why we use the metric system. And as long as we know the basic units, we just have to know the prefixes that I've written on the board. And then to do conversions from like one unit to the other, if we want to go bigger or if we want to go smaller, we only have to do multiply by 10 or divide by 10, okay? It's very easy. We're either dividing it by 10 or we're multiplying it by 10, and then that simplifies it for you, okay? But for example, if we think about a conversion between centimeter and inches, feet and yard and all that, these numbers can get pretty complicated. So you won't be able to do it, right? Like if I ask you go from centimeters to inches, it's complicated, right? So not everyone can just do those conversions, and it can get difficult to keep track of all of that. So for the metric system, it's very easy and it's simplified. You just have to remember the prefixes, and then that's on the board. And then you have to remember if you're going to multiply it by 10 or divide it by 10. Okay? So the prefixes on the board is the ones that I have here in red in your slide. And then basically what we're doing, everyone, is you're going to your right, we're gonna multiply by 10. And then if we're going to the left, then we're going to divide by 10, okay? And then it's not like to the right of the base, to the left of the base. No, for example, if I'm gonna go from centimeters to millimeters, right? I'm gonna multiply by. So I'm gonna go from centimeters to millimeters, I'm gonna multiply by 10, right? Or if I'm gonna go from centimeters to decimeters, I'm going to divide by 10, right? So if I'm going in that direction, and then each step is 10, if I need to jump 2 steps, then it becomes another 10, right? So 10 times 10 would be 100, right? If it's 3 steps, then it's 1,000, and so on. So yeah, everyone, it's the metric system. It simplifies it. It makes it easier. So if we're gonna go towards a bigger unit, then we have to divide it by 10. If we have to go towards smaller units, then we have to multiply by 10. Okay, and then we'll do some examples together so that we'll feel more comfortable about it. Actually, let's do one. For example, I have 1 meter, and then I want to know how many centimeters is that. Okay, 1 meter to centimeters. So everyone remember that the base can be either meter, liter, or grams, depending on what we're talking about. This example that I'm asking you, it's in meters. We'll do one example of each, but they're all the same. Okay, I want to go from meter, which is the base, to centimeter, which is here. So I'm going 1 times plus 10, and then another 1 times 10, right? 10 times 10 is 100, right? So I'm going times 100. So 1 times 100 is 100, right? So 1 meter is 100 centimeters, okay? Or let's go from, let's say I have 1 kilograms, and then I want to know how many grams it is, okay? This is kilograms, and then this is grams, okay? So how many times am I? So this is 1, right? Times 10. This is 2 times 10, and this is 3 times 10. So 10 times 10 times 10, I'm multiplying by I heard 100, but that's not what I was looking for. 1,000, right? There's 3 zeros. Good, good, good. So 1 times 1,000 is 1,000 grams, okay? And the opposite is true. If I have 1,000 grams, right? Or let's make it a little bit different. If I have 1,000 grams, and if I want to know how many kilograms is that, okay? Now we have to do the opposite. So I have 10,000 grams. The grams is here. And then I have to go to kilograms, okay? So how many times am I going back? 1. And then because I'm going to the left, what am I doing? Division or multiplication? Dividing, right? Divided by 10. And then 1 more divided by 10. So we are dividing by 1,000, right? 10 times 10 times 10 divided by 1,000. And then everyone, this is how I do it. When it comes to division, you can just use your calculators, or just do it in your just 10,000 divided by 1,000, and then you just cancel the zeros, right? That's one way to do it. And then you cancel these zeros, right? And then or another thing, if it gets more complicated, everyone, you can do 10,000.0, okay? By the way, this is an easy one, but let's say if the numbers were more complicated, and then we have to go 1,000 times to the left, right? 1, 2, 3. So we move the decimal point inside, and then it's 10.0, which is 10, right? It's 10 kilograms, okay? And then this is an easy one. We can do like the one I showed you, but whenever there's a decimal point, or whenever there's no decimal point and we have to do a division, I just add 0.0 at the end, and then I just move it inside however many times I need, okay? And then if we run out of numbers, you start adding 0.000, whatever number it is, okay? And then we're gonna do some examples together that I want you to do it. If you have a scratch paper or something, follow along with me and let's do some examples together, okay? But for us to be able to do all of this, if we remember these prefixes, kilo, hecta, deca, and then the base, and then deci, centi, milli. It doesn't matter if it's for liters, grams, or meters, right? We just have to go left or right, and then just remember, if you go to the right, it's going to be times 10. If you're going to the left, it's going to be divided by 10. Okay, so we just have to, if we just remember the prefixes and know division and multiplication, right or left, then we should be good, okay? regardless of whatever the base unit it is. I'm going to give you an example. Let's do this together. Let's say you have 7 grams and you want to know how many milligrams is that. Okay, 7 grams. We want to know how many milligrams is that. 7 grams is how many milligrams? I'll wait like a couple of more seconds and I'll ask the classroom to just say the answer so that we'll give everyone a chance to do the change. Are we ready? Some are ready, some are we all ready, everyone? Did you get the answer? Okay, how many milligrams? 7,000 milligrams, right? That's correct. Okay, so basically you're here and then just going 1, 2, and 3, right? So times 1,000, 7,000 milligrams. The next number is, we're gonna do, let's do volume, 3,400 milliliters to liters. 3,400 milliliters to liters. So from milli to liters. Can use the method I showed you. You can add 0.0 and then just move the decimal point inside, right? Hard. I don't think milliliters to liters. Yeah, milliliters to liters. Okay, so we're going the other way, right? Divided by 10, divided by 10, divided by 10. So it's divided by 1,000, right? So 3,400 divided by 1,000. You can do this way. This is our decimal point. You can just add 0.0, 1, 2, and 3. We'll put the decimal point here. So what's the answer, everyone? 3.4 liters. Did you get that answer? Did you get that number? All right, good, good, good. Okay, and then the last one, let's do something in centimeters. Okay, we have 53,000 centimeters. We want to convert it to kilometers. Centimeters to kilometers. That's a long way. I think some people got it, but some people are doing it, so I'm gonna wait. So how many times do we move the decimal point and which direction? Which direction are we going, left or right? Left, and then how many jumps? How many times? 5 times, right? Good job. Okay, so centimeters to kilo, that's 1, 2, 3, 4, and 5. Okay, five zeros is 1, 2. That is 100,000, right? So 53,000.0. We're gonna move it 5 times. 1, 2, 3, 4, and then 5. So this is where we're gonna put the decimal point. So what do I add here? 0. What's the answer? 0.53 kilometers, okay? Did you get it right? Okay, good. And then in case you didn't get it right, do you understand now how to do it? All right, good, good, good. All right, so I won't ask this on the exam, but it's good to know because it's gonna come back in physiology next semester, okay? And then I do go over this in physio when I teach it, and you are gonna need this information. Some people do some conversions in this way, you know, but I like the way I use the way that I showed you on the board. If you feel comfortable, you can choose this method or just do the one that I shared with you, okay? Everyone, any questions about the metric system? All right, okay. So now that we're done with lab 1, we're going to start lab 2, okay? Everyone, lab 2 is, so we're gonna cover lab 2. Lab 2 has a lot of information, but not information that you've never seen before. It's kind of like a review from Bio 5, okay? And the only thing is, these labs, this takes like 4 or 5 labs in Bio 5, but we're gonna do it in 1 lab in anatomy, okay? And then a lot of the stuff, it might take some time just because a lot of the material, you know all about it, you've studied, you've taken the test, but in case you haven't understood the material or, okay, this is this, but why, right? And all that, I wanna cover that material as well. And whatever we're covering in lab 2 today, I cover more in details in chapter 3 lecture recording, okay? So this week you have chapter 2 and chapter 3 in lecture. I don't know if you got the chance to look at it, but chapter 2 does not have a lecture recording. Lecture 3 is very long, okay? So lecture 3 is enough time for like 2 chapters, okay? Chapter 2 has a slide deck, and chapter 2 has a handout for extra credit. So for chapter 2, you're supposed to review the slides. Sorry, you're supposed to review the slides. Also, if you have, if some things are not clear, there's also additional resources like short YouTube videos for chapter 2. And then please complete the handout and turn it in. It's 10 points extra credit, okay? So far, usually in the spring semester, you get between like 30 to 40 points extra credit, and then this 10 points is one of them, okay? So make sure that you turn it in, or if you have any questions, bring your questions to me during office hours or email, or ask me before or after class, okay? Make sure that you review the slides and then you complete it, okay? And chapter 3 is long, and then whenever you're watching chapter 3 and you feel like it's long, just remind yourself that you don't have to watch chapter 2, and chapter 3 is long, so it just makes it up, okay? And then it's gonna be, has anyone watched chapter 3? Oh, wow, good. The whole thing, it was long? Yes. Did you do it in one sitting or maybe no, 2, 3 times? Yeah. It is long, okay? So today this is gonna be easy for you because you've watched it, and then whoever, if you haven't watched it yet, I mean, it's okay, it's still Wednesday, probably you're working on chapter 2. I'm just gonna say that and think you're working on chapter 2. But after this, when you watch chapter 3, you will understand it better, okay? But make sure that you're paying attention and you're watching the videos. Otherwise, you won't be able to answer to the exam questions, okay? Lecture exam questions are all conceptual questions, understanding and participating, okay? Yeah, so in today's lab, everyone, we're gonna cover the cell, the structure and the function of the cell. And then we're gonna talk about transcription and translation. We're gonna talk about mitosis and meiosis, and we're gonna talk about how to use a microscope, okay? Introduction to microscopy. The last part is easy because I'm pretty sure if you did in-person lab for your Bio 5, you've done labs where you've used the microscope, okay? But we just have to review it again and do 2 small activities on the microscope today in lab, just because soon we're gonna cover the tissues, and then we're gonna look at a lot of slides under the microscope using the tissues. And then some of us, we probably haven't used the microscope since last semester, right? So it would be good to get back at it, right? And use a microscope. So everyone, we're gonna start with lab 2, and please let me know if you have any questions, okay? And then if you get the chance, if you want, you can take a picture of the whiteboard with the metric system, because soon I'm gonna need to erase it so I can write more things, okay? So feel free to take pictures if you need to. All right, so let's start the lab. So everyone, first I wanna start by saying that all living things are made up of cells, okay? Any living thing is made up of cells, from bacteria, humans, animals, plants, anything that is a living thing, it's made up of cells, okay? And then we have 2 types of cells. We have, does anybody want to share what are the 2 types of cells that we have? Eukaryotic cells, and we have what? Prokaryotic cells, right? Does anybody want to share what's the difference between a eukaryotic cell and a prokaryotic cell? Like one major main difference between the 2? The nucleus. Yes. That's correct. Thank you everyone for participating. Please participate, whether if it's right or wrong, like just participate, and then so that we can learn together, okay? I appreciate everyone who's participating. So we have eukaryotic cell and prokaryotic cells, and then the main difference, yes, there are other differences, but the main difference is eukaryote. The term eukaryote means through nucleus. So the eukaryotic organisms, they have a nucleus, and whereas prokaryotic cells, they do not have a nucleus, okay? And then the nucleus, everyone, is this, I don't know, is it purple? Is this purple ball that you can see inside of the cell, okay? And eukaryotic cells have nucleus. And then examples of eukaryotic cells are human beings. Animals are made up of eukaryotic cells. Plants are eukaryotes, right? And then examples of prokaryotes, for example, we have unicellular prokaryotes, for example, bacteria. Bacteria are made up of only one cell. One cell makes up the whole bacteria, right? And it doesn't have a nucleus. And then speaking of nucleus, why is the nucleus important? Like, what's inside the nucleus? DNA, right? And then our DNA, our genetic information, the information that makes us who we are. It gives us all of our characteristics and everything that we are, all of our genes, our genetic information, right? It's in our DNA. And then that DNA in the form of chromosomes or chromatin, we'll talk about the difference. They're all enclosed, enveloped, and protected inside of the nucleus, right? And whereas for prokaryotic cells, they have the cell, and then their DNA would be just inside of the cell, like it's not housed inside a nucleus, okay? And the nucleus is important because it contains our genetic information, okay? And eukaryotic cells, usually eukaryotic cells, the ones that have nucleus, they are made up of they are multicellular organisms. Like, for example, us humans, we are eukaryotic organism, and we're made up of billions and billions of cells, right? Animals are made up of many cells. Plants are made up of many cells. And this is just an interesting fact. It has nothing to do with anatomy. Actually, sometimes human beings, they have more bacterial cells on us than our actual own cells, okay? It's either part of our, microbiota or microbiome, or it's just, even when we shower, we still have like bacteria, right? So we have more bacterial cells than our actual cells. That's something I covered in micro. Anyways, but we are multicellular, okay? And everyone, we have different types of cells that we're gonna see in the next slide. For example, our nerve cells. Do you remember what a nerve cell looks like? Do you remember the dendrites and the axon, right? Nerve cells look different than muscle cells. Remember muscle cells, maybe from last semester with the actin and the myosin, right? It has striations. It looks different, right? Or skin cell, like an epithelial cell, it looks different than a nerve cell. Or a blood cell looks different than everything else, all the other cells, right? So we have diverse types of cells, but they're all our cells. And then all of these cells, except for the red blood cells, all of our cells, they have a nucleus. And then inside the nucleus, they have the same genetic information, which is our DNA, which is found in all of our cells, okay? The only one that does not have a nucleus is going to be the red blood cells. And then we'll come back to this slide. Like here, for example, if you look at it, the nerve cell, do you see the nucleus in the nerve cell, right? Do you see the nucleus in the epithelial cell, right? And we also have, these are the purple dots are the nuclei in the skeletal muscle. And then you see how those are all our cells, right? We're made up of these cells, but they look different. And then the reason why they look different is because of the structure defines the function, right? Because of the purpose that it serves. Like our nerve cells, they have to look like this so that they can transfer the message from one nerve cell to the other cell through the dendrites and the axons, right? During action potential. Or the skeletal muscles, do you see these striations? It's for relaxation and contraction, right? When we're moving our muscles, correct? That's why the skeletal muscle looks like that. Or the epithelial cell looks like that. And then we also have the red blood cell, which also looks different than other cells. And then it's an exception. Red blood cells don't have a nucleus, okay? But the nucleus that we see in all of our other cells, it has the same genetic information, each person's own DNA, which makes you who you are. It's housed inside your nucleus, okay? Everyone, here you can see a eukaryotic cell. So we talked about it. There's the nucleus there. Inside the nucleus, there's going to be the DNA. And then obviously the cell is protected by, what's that outside layer that protects the cell? You can see it in the picture. What is it called? The membrane that protects the cell? The plasma membrane, right? And then inside of the plasma membrane, inside we have the cytoplasm, which is inside of the cell. It's filled with cytosol, which is the liquid, and we have organelles. So our cells, it has the nucleus with our genetic information, and outside of the nucleus, which is still inside of the cell, there's this like factory or full of machinery or organelles that their job is to serve our cells, right? To either go through mitosis, meiosis, to make more cells, right? To go through cell division, or to make more proteins through transcription and translation, right? All of our genes that we have in our DNA, which is inside our nucleus, they have to, for these genes to be expressed, it needs to go through transcription and translation, okay? And then, are we all familiar with transcription and translation? Like, do you remember a little bit, right? I mean, we're gonna go in details and we're gonna review it, but you do remember a little bit what transcription and translation is, right? So transcription and translation is important because in our DNA, we have our genes, our genetic information, right? And then if you look at the DNA, it's made up of these double helix. And then do you remember how there's these letters, the A, T, G, C letters, right? These letters are codes, and then these letters, they come together and then they make our genetic information. But just as letter in our DNA, they don't serve anything, right? They have to be transcribed to messenger RNA, right? And then they have to be translated, right? Translation means protein synthesis so that proteins are made, and that's how our genes are expressed, okay? Most of the stuff in our body, all of the molecules and all the organelles, everything that works in our body is mostly made up of proteins. And then for the body to make these proteins, it needs to express those genes in our DNA, okay? It needs to go through transcription, translation, and make these proteins so that we can have all the molecules or everything that we need in our body that our body makes, okay? So that's what we're gonna talk about soon after we review the organelles. But everyone, just because we're looking at this picture, and then I'm jumping ahead and I'm sharing a lot of information, but don't worry, this is just introduction where everything I'm saying, I'm gonna go into details, okay? What you're seeing here is the nucleus. The nucleus has our DNA. During transcription, our DNA cannot leave the nucleus in the form of DNA, so we have to transcribe it to messenger RNA. And then we'll talk about that. You probably, like we said, the letters ATGC, we just have to convert it from DNA to RNA. We'll review how to do that, right? The G binds to C, the A binds to T in DNA, but it binds to U in RNA, right? And then we'll review all of this. I'm sure you know this, but we'll review it together. And then once the DNA leaves the nucleus through the nuclear pore, you can see outside here. Can anyone tell me what is this over here? What do we have here? What is this structure here? It's the rough endoplasmic reticulum, okay? And then how do we know that it's the rough one? Because we have rough endoplasmic reticulum and then we have smooth endoplasmic reticulum. The rough one is the one that has these dots on it. And then these dots are the ribosomes, okay? And then ribosomes are where translation takes place. That's where protein synthesis happens, okay? So just imagine the DNA leaves the nucleus in a form of messenger RNA, and then it leaves the nucleus, it goes to ribosomes. Those are all ribosomes. And then in the ribosomes, that's where the protein is made, okay? And then what are proteins made up of? What makes a protein? Or if we want to do or if we break down the protein, it gets broken down into what is a simple form of protein? Amino acids, right? Yeah, so on the ribosomes, we'll see like a bunch of amino acids coming together and making a protein, okay? And then after the protein is made, it leaves, it goes through the, this is the Golgi apparatus where the protein gets sorted and everything that it needs to go through. And then it goes to its destination, whatever protein it is, whatever function it has to do, that's where it goes, okay? Everyone, this is just a general overview, okay? This is just a general overview of what we're gonna talk about in details, but I just wanted you to see the bigger picture, what happens in the nucleus, how the DNA leaves, it goes to the ribosomes, and how protein is synthesized, okay? And then all of this is happening so that our DNA, the code, the genetic code, those letters on our DNA, they have to get transcribed and then translated, and then that's how our genes gets expressed, okay? So that's what we're gonna talk about more in details. But first, we're gonna review the organelles. What are some organelles here that you're familiar, that you wanna share with us, like from this picture? Mitochondria, right? Do we all see the mitochondria, right? The mitochondria, what's the function of the mitochondria? What's the nickname for the mitochondria? What is it known for? The powerhouse. Why is it called the powerhouse? Yes, it generates, right? So we have different phases where the body generates ATP, which is the form of energy, but most of our energy is generated in the mitochondria. But we're not gonna go more in details about that, okay? But the reason why we call it, because if you look at the amount of ATP built, right, made, most of the ATP is generated inside the mitochondria. Do you see any other organelles that you're familiar with? Anything do you see familiar that can serve in cell division, mitosis, meiosis? Maybe it builds the spindle fibers where the chromosomes line up on. Centrosomes, right? The centrioles up here. All right, yeah. So those are some things that we're gonna go over, okay? So are we okay on this slide, everyone, with the different types of cells that we have, right? Good. Oh, speaking of our plasma membrane, right? Our plasma membrane, so each cell has a plasma membrane, and then obviously we do need something to protect the cell, right? The cell needs to be protected because it has all of our organelles, it has the nucleus, it has our genetic information, it needs to be protected where to a certain point where not anything can enter the cell or not anything can just leave the cell, right? It needs to be regulated. So our plasma membrane, everyone, is made up of phospholipid bilayer. And then when we say phospholipid, here we can see like this is one layer. So it's made up of the head and the tails. And then when I say bilayer, basically you'll see the head and the tail, and then another head and the tail like this, okay? That's how bilayer, bi means two layers, okay? So that's how we have the layers, but the layers are like the bottom one is inverted, okay? Where the two tails come face each other and then the heads are outside. And then the reason for that is our phospholipid bilayer is made up of this polar head, okay, which is hydrophilic. Everyone help me understand this term. Hydro stands for water. Philic means loving, okay? So the outside portions, right, the heads, are they okay with touching the water, getting in contact with water? Yes. And then the tails are non-polar. It means they are hydrophobic. Hydro means water, phobic, phobia, fear, right? They don't like to be in contact with water. That's why whenever we have the phospholipid bilayer, you'll see how the tails are facing each other and then the heads are outside, okay? So here in this image, we can see we've cut out a small section of the cell with the plasma membrane, and then we've magnified it here. And then this is only like a small section of the cell, okay? Everyone, like this is supposed to be a huge cell, and then this is inside of the cell. This is the plasma membrane, and then where you see extracellular fluid, the ECF, that's supposed to be the outside of the cell, okay? So, and then this is the plasma membrane, and then we're talking about the plasma membrane, how it protects and not anything can just get inside the cell or leave the cell, and it's highly regulated, right? And then as it should be, because we have a lot of precious things inside of our cells, right? It makes us who we are. And then everyone, if you look at the plasma membrane, do you all see the phospholipid bilayer, like the tails and the head, right? That's good. And then one important characteristic about our plasma membrane is semi-permeable. Semi-permeable means it allows things to pass. Semi means it allows certain things to pass through the membrane, but not everything, right? For example, looking at this image, we can tell that if something is like very, very, very tiny molecule, it will probably slip between the pores and enter, right? But for example, if we're talking about bigger molecules, they can't just fit through these pores and enter the cell. That's why we have special transport proteins, which I'm not going to go into details. I'm just going to introduce it to you. Those are, we have different types of proteins called peripheral proteins, integral proteins, or protein channels, right? Where, you know, it allows molecules or ions or pumps, right, to get inside the cell or leave the cell, okay? And then you can see we have some cholesterol inside the membrane. So our membrane, it's not super hard, it's flexible, and then having cholesterol, like some lipid inside, gives it that flexibility. Like it's not hard, right? Fat makes it a little bit flexible. And we also have some carbohydrate chains inside of it. Okay, so this is just an introduction to our plasma membrane. And then here, everyone, we can see the cell and all of the organelles. We did mention some of these organelles, but not everything. And then we're going to go over each and every one of those one by one. And there is a summary table that you can review each organelle with the function, okay? And then you have seen this in Bio 5, but maybe not like in details or just random, you know. So the ones that you're not sure, you'll get to learn again and understand it this semester, okay? So everyone, like we said, we have the nucleus. So inside the nucleus, we have the nucleolus. And then, but inside the main nucleus, we have our DNA. Here in this picture, our DNA is in the form of chromatin. Shortly, we're going to see what DNA looks like and why it looks like the way it looks like, or why do we draw it like an X, right? So if I ask you to draw a chromosome, we draw it like an X. like where does that X come from? And here it's in the form of a chromatin. Chromatin is, it's still like, it looks like threads, okay, just threads. It doesn't look like X, or it doesn't look like the DNA double helix that it's supposed to look like, okay? So it's in the form of a chromatin. We have the nuclear envelope, and then we have nuclear pores. So you know how we said that the DNA cannot leave the nucleus in the form of DNA, it has to be transcribed to messenger RNA, right? So it will leave through these nuclear pores. And then after it leaves through the nuclear pores, now it's in the cytoplasm, it's gonna go to the rough endoplasmic reticulum, which is this. And then on the rough endoplasmic reticulum, what are these dots that we have? Yes, where's the label? Yeah, ribosomes. Okay, so these dots are ribosomes, and then the messenger RNA is gonna travel to the ribosomes, and then we'll see an image of the ribosomes. And it doesn't look like these dots, it looks like two subunits, the small and the large subunit, where translation takes place and protein is synthesized. And then it passes through the smooth endoplasmic reticulum, and then passes through the Golgi apparatus, where the protein gets sorted. And then some other organelles that we have, you mentioned the mitochondria. We have lysosome and peroxisome. So with all of this happening in the body, we are gonna need a person for, I don't know, to recycle, to repair, get rid of dead organelles, get rid of dead enzymes, right? So those will do that. And then we have, and then up there, we have, do you see the two centrosomes with centrioles, right? We'll learn today when we review mitosis, meiosis, right? Whenever we have cell division, these centrosomes, they travel on opposite sides of the cell. They build these spindle fibers, right? And then that's where the chromosomes, they line up on the equator, okay? And then you'll remember it when we talk about prophase, metaphase, anaphase, telophase, the phases. But it's involved in cell division. Okay, so this is just magnifying parts of the image. We talked about those. Okay, so this one is reviewing each organelle one by one. So mitochondria, we said the mitochondria is the powerhouse. Most of the energy ATP is generated there. So remember on the rough endoplasmic reticulum, and then I said the reason why it's the rough endoplasmic reticulum is because it has yeah, you guys can close the door if you want to, okay? You get to choose the row over there. You choose if you want. Please close the door. We have the ribosomes. The ribosomes are found on the rough endoplasmic reticulum. Remember, we had rough ER, smooth ER, right? The smooth ER does not have the smooth endoplasmic reticulum, doesn't have ribosomes. Rough endoplasmic reticulum has the ribosomes. And then those dots are the ribosomes. And then this is what ribosomes actually looks like. They're made up of small subunit and large subunit. And then I don't know if you remember how translation takes place. Do you remember like a tRNA comes here, and then there's a messenger RNA with 3 letters, which is a codon, and then an anticodon, and then the tRNA brings an amino acid, right? Anyways, we're gonna talk about that, but this is the ribosome, and that's where translation takes place. And then when we talk about translation, you'll remember that this is the ribosome that we're talking about, okay? So translation takes place in the ribosomes, okay? And then this is the ribosomes. The next image, everyone, is showing us the endoplasmic reticulum. We have rough endoplasmic reticulum, and then we have smooth endoplasmic reticulum. Do you all see the difference between the two, right? One has ribosomes, and the other one doesn't have ribosomes, right? And then basically the job is to produce proteins that the body needs so that it can do the function of the cell, okay? We also have the Golgi apparatus, and then this one modifies, packages, sorts proteins and other products that was made by the endoplasmic reticulum. And then everyone, the ones that we covered so far or we reviewed, this is a summary of the organelles. So we have the mitochondria. I'm not going to read the structure. I'm just going to read each organelle and the function, the last column. So like we said, the mitochondria, it synthesizes the majority of the cell's ATP. Peroxisomes, they detoxify certain chemicals, and ribosomes, they synthesize proteins. Rough endoplasmic reticulum, which contains the ribosomes. The rough endoplasmic reticulum modifies and folds proteins made by the ribosomes. We have the smooth endoplasmic reticulum. Smooth endoplasmic reticulum is a little bit different. It stores calcium and synthesizes lipids. Golgi apparatus, it sorts, modifies, and packages the proteins that were made by the endoplasmic reticulum. Finally, the lysosome. Lysosome is, you know, let's say we have damaged organelles, right? We need someone to digest those damaged organelles. So that's the job of the lysosome, okay? And then it recycles and it collects the damaged organelles. We also talked about the centrosomes with centrioles. So the centrosomes, we're going to see that they're involved in cell division. And basically, before cell division, the centrosomes, they duplicate and then they go on opposite ends. And then the centrioles are the ones that they make the spindle fibers, where we're going to see those threads where the chromosomes, they hang up on during, line up on during cell division. Now we're going to talk about different types of cells, okay? Everyone, we have cells, but we have specialized cells. It means that these cells, they're special. They have something special. For example, microvilli. Some cells have microvilli. Those are the cells that I'm going to call these, the stuff that you're seeing on top of the cells, I'm going to call this a brush, okay? Because it looks like a brush. Do you see that, right? These microvilli, I'm going to call it a brush. Microvilli is a specialized cell. And then remember we said that some cells, they're made a certain way. They have the structure that they do because of the purpose or the function that they're going to serve, right? So everyone, this microvilli, the main purpose, if you're taking notes, the main purpose for microvilli is to increase the surface area for absorption. What does it mean, increase surface area of absorption? So you see how here, the part where it has the brushes, like it goes up and down, up and down, up and down, up and down, right? All of these, every time that we go up and down, all of these are made up of cells, and then the job of these cells is to do absorption, okay? And if I basically just imagine that you have a long line of cells, but they look very compact because they are in the form of microvilli. It means they go up and down. So basically, it has a large surface area for absorption, but it looks very compact, right? An example of one of our organs that has microvilli is our small intestine. So whenever we eat our food, it goes down the esophagus, stomach, and then to the small intestine. In the small intestine, we have like further digestion happening, but also small intestine is the large destination where whatever nutrients we have, that is going to get absorbed, right? Because what's after small intestine? Large intestine. And then what goes to the large intestine? Whatever needs to be discarded, right? So small intestine is the large destination where all the nutrients from the digested food gets absorbed. And then guess what? The small intestine is made up of microvilli. The cells of the small intestine is made up of microvilli, and small intestine has a large surface area for absorption. And we need that because why did we eat all that food? And it went through digestion so that we can get all the nutrients, right? So the small intestine is going to absorb it. Another special type of cell, everyone, is the cilia and the flagella. The cilia looks like the flagella, but I called the cilia looks like the microvilli, not flagella. It looks like the microvilli, but microvilli, I said it looks more like a brush. The cilia, it looks more like this hair-like structure. And then also whenever the cilia moves, it also has this sweep-like structure as well. Okay, this is how the cilia moves. And then again, just like the small intestine and the microvilli, we also have special cells that have this cilia, this hair-like structure on top of it because of the function. Okay, For example, let's say you have something stuck in your throat, your trachea, and then you want to let it out. What do you start doing? Coughing, right? And then whenever you're coughing, you want something in your trachea to like do this sweep-like motion, right? To push whatever you have stuck in your throat up, right? So for example, our trachea, our respiratory tract, it's made up of specialized cells like cilia, and then cilia does this hair-like or sweep-like motion where when we're coughing, it helps us let it out, okay? That's an example of the cilia. Here, this is an actual picture of like a cell with cilia under scanning electron microscope. It really does have that hair-like structure. And then this is the function. This is how it moves. It has this sweep-like structure, okay? Another special cell, another thing that special cells have is a flagella, okay? And then what is a famous cell that has a flagella and without the flagella cannot do its job? The sperm cell, right? So the sperm cell has a flagella. It helps to swim and get to its destination. And then without flagella, it cannot do it. And then the reason why the sperm has a flagella is so that it can serve its purpose, right? Do its job, just like all the other cells that we mentioned. Okay, back to organelles. We have the nucleus, right? The nucleus has the nuclear pore. That's where the DNA in the form of messenger RNA leaves the nucleus, okay? Any questions about organelles, anyone? Okay, so next I want to talk about protein because of a mutation or a change in the DNA, okay? So that's how important it is, the DNA sequences and the genes on the DNA, right? Because those are the letters that go through transcription, translation, and make the proteins that we make, okay? Everyone, this is the central dogma. Whoever's taking bio classes, you have to know this, like memorize this, or you already know it. I'm just like bringing it to your attention. This is the central dogma, everyone. So your DNA gets transcribed into messenger RNA, and then that messenger RNA gets translated into protein, okay? So the DNA is the DNA that has the sequence, the genes, the letters, and then the DNA cannot leave the nucleus in the form of DNA, so we have to change it into, from DNA to RNA, and then it's called messenger RNA, right? Because it's the messenger, it's taking the DNA code so that we can translate that code and make a bunch of amino acids, which are gonna come together and build what? Proteins, okay? And where does translation take place? Ribosomes. Yes, ribosomes. Good, good, good. All right. So everyone, this is what I was talking about. So you have your DNA code. It gets transcribed to messenger RNA. And then once you have your messenger RNA, every 3 letters are going to be called a codon. Okay? You probably remember an activity or exercise that you've done in Bio 5. Like you have to take the 3 letters and find the anticodon and then see that 3 letters it matches with which amino acid, right? If not, we'll review it. So in this table, everyone, what you're seeing is, so we said that the DNA sequences, these letters, the ATGC on the DNA is very important because they're gonna get transcribed to messenger RNA. And then every 3 letter is gonna be a codon where the anti-codon is gonna code for an amino acid. So what we're seeing here is we have 20 different types of amino acid, and then all of our proteins are made with this 20 amino acids, okay? But for example, each one can be used several times, like there's different, there's different lineups, like different, what's the word? There's a word I'm trying to say. Like there's different conformation of, no, that's not the word. So there's different ways that we can use these 20 proteins, like put them together so that we can make all the different proteins that we need to make. Okay, different arrangements. Is that what I'm trying to say? Anyways, but the good news is, everyone, if you can see, there are some letters that if you change like one letter, they'll still give you the same amino acid. Okay, that's really good. For example, look here. GCU, GCC, GCA, GCG, okay? All of these gives us the amino acid alanine. That's good because if we have a mutation in the 3rd row, right? No matter if it ends up being the U, C, A, and G, it's still gonna code for alanine, which is the right amino acid, and then we're gonna make the right protein, right? But let's say instead of GCU, the C mutated to an A. Now I have GAU, right? Does GAU code for alanine, or it codes for aspartic acid, something else, right? So now that's a completely wrong amino acid, which leads to the wrong structure of protein. Okay, so yeah, so that's one thing that, whenever we have different types of mutations, either substitution, like different letters, sometimes we have deletion. So if one letter is not there, then the 3rd letter is gonna be the 1st letter of the next codon, right? So the whole thing is gonna change. And then, yeah, sometimes those not only lead to like abnormal structures of protein, but also, for example, for translation to start, we need the start codon. The start codon is AUG, which is methionine amino acid, which is the start codon, right? If it doesn't hit AUG, then we can't even start making the protein, right? Or when does the body knows that it needs to stop translation? When it hits what? What codons do we have here in red? Those are the stop codons, right? So whenever it hits UAA or UAG or UGA, those are the stop codons. It means that translation is over, release the amino acid, now the protein is built, right? Imagine if one letter is wrong there, and then it doesn't hit the stop codon, and then it keeps translating, it doesn't stop, like, right? That's how we end up making these abnormal structures of proteins, okay? But what I want you to understand here is that so all of this, for us to be able to decide which amino acid, what is the anticodon, it all comes from the sequence of the DNA, right? That gets transcribed to messenger RNA, and then which is each 3 letters makes a codon, and then we come up with the anticodon, like RNA to RNA, and then these letters would be the anticodon, and then we would check this table, this anticodon or this codon, which amino acid covers these 3 letters. Okay. Another thing, I won't ask about this, but I want to cover it so you know about it. So everyone, I kept talking about all of this comes back to our DNA sequence, right? The letters that we have around DNA, the ATGC. So, imagine the sequence on your DNA that we're seeing, all these letters, okay? This sequence, they have coding regions, and then they have non-coding regions. It means that we have areas on our DNA sequence that the letters mean something, they code for a gene, they're meaningful, and then we have sections on our DNA that it doesn't code for anything. Like they're just there, okay? So those non-coding regions, okay, those are called introns, the purple side, I mean the pink ones. And then the coding sequences are called exons, okay? Exons are coding sequences, introns are non-coding sequences. And then during transcription, what the cell has to do is it needs to get rid of the non-coding sequences, right? What do we need? We just need letters, right? And then we just need letters for our messenger RNA so that we can do translation. So basically, everyone, pre-messenger RNA transcript. So basically, before we get our final messenger RNA, which is going to be only exons, right? And then here they change the color. So the introns are the yellow ones, and then the red ones are the exons. So here during transcription, what happens is everyone, the introns, the non-coding regions, like the regions that they don't code for anything, they get spliced up, and then whatever is left, those are called the exons. These are all coding regions. Like every single letter here, it means something. And then the exons, they come together. This is a whole coding regions, all letters that mean something. And then we have our messenger RNA, okay? So I won't ask about this in lab, okay? But I wanted you to be familiar with like our DNA. Yes, it's made up of the nitrogenous bases. The DNA sequence has all the letters for our genes. But on our DNA, we also have part of the sequences that they don't code for anything, like they need to be removed, okay? And then the exons are the coding region. By the way, there's a mutation or there's a condition that I just learned about. I'm not going to go into details because we don't have to know. It's not important. There's a condition where now that, it's the time where we can do genetic tests and all that stuff, where people might be missing like an exon, right? Like on let's say on chromosome number 9, exon number 8, for example, which is a coding sequence, right? They might be missing that sequence, right? And then that person would be lacking in whatever that sequence codes for, because they don't even have the what's it called? The code, right? The sequence, so that it goes through transcription and translation. So and then exons are important, right? They are coding regions. They're there because they're responsible for something. Just wanted to share that. Okay, now let's talk about transcription. We kept talking about transcription, translation, transcription, translation. We're gonna go more in details. Actually, not super details. Basically, I'm gonna magnify this image so we can see it clearly, but I want to show you the big image here. So all the way at the top, you can see that there's a promoter region. That's where our RNA polymerase binds. And then we need enzymes for transcription to happen, right? And enzymes are very important. So wherever we have the promoter region in the first image, you see in the second image, the RNA polymerase, the enzyme comes. And then remember the DNA double helix, it unwinds the DNA double helix. Do you see that? The DNA strand, it opens up, right? Now we have the leading strand and the lagging strand. Basically, this is the DNA strand on one side, and then this is the DNA strand on the other side. This was a double helix. The RNA polymerase, it opens it up, and then it uses this as a template of a DNA strand, these letters here, and then we go from DNA to RNA, right? The G's bind to C's, the C's bind to G, the A's bind to U, right? And then it makes this template here, which is gonna be our messenger RNA transcript, okay? And then that's how our DNA information in the form of messenger RNA, it leaves our nucleus. Now this messenger RNA is gonna go to the ribosomes, right? Because each 3 letters for the messenger RNA is a codon that codes for an amino acid, okay? But let me just magnify it. This is how transcription starts. So we have this promoter region, and then with a transcription factor, it means that that's the part of the gene that we want to transcribe and express. And then, so that's where it initiates transcription. RNA polymerase comes in. it unwinds the DNA double helix so that we can use one side of the DNA as a template. And then it opens it up. And then as you can see here, it's using this side of the strand as a template. And then you can see how it's adding complementary bases from DNA to RNA. And then it's making the messenger RNA transcript. And then this is where it also gets rid of the introns, and then it brings all the exons, the coding regions together. And then termination is when it ends, and then it releases the messenger RNA, right? And then do you know how we learn in Bio5? Oh, DNA is double-stranded, RNA is single-stranded, right? So this is RNA single-stranded, right? So now this messenger RNA is gonna go find the ribosomes, right? So that it can go through translation, okay? And then remember the ribosome here, right? This is the ribosomes. It's made up of small subunit and large subunit. The messenger RNA is gonna come, and then you're gonna see the messenger RNA here, and then we're gonna see each 3 letters. It's gonna make a codon. And over here, we're gonna see tRNA, transfer RNA. It's gonna come with an anticodon, and it's gonna bring the complementary amino acid for that codon, okay? So let's go there to see it. So everyone, this one is, okay, if you look really hard, doesn't this look like the letter T? Right? Okay, so that's tRNA, right? And then it is made up of RNA strands, by the way. It's the tRNA. The T stands for transfer RNA because it's transferring. For example, mRNA is the messenger RNA because it's the messenger. It's transcribing the DNA to RNA and it's bringing the message, right? And then those 2 ribosomal subunits that we saw, those are rRNA. It means ribosomal RNA, right? And then that's where translation is going to take place, okay? So everyone, what you're seeing here is, remember I said on the small subunit, we're gonna have the messenger RNA, and then the first 3 letters, they're gonna be a codon, right? And then they're gonna have an anticodon. Anticodon is literally everyone just RNA to RNA, okay? So if this is AAG, it means here we had UUC, the anticodon is AAG, okay? And then AAG, it codes for phenylalanine. So this is our tRNA bringing in the amino acid phenylalanine. Okay, and then that's how it's gonna go. So here, again, this is not the actual picture of translation, but here we can learn how, remember, once upon a time, we had our DNA strands, right? The letters on our DNA, which got transcribed into messenger RNA, right? The TAG, it turned into AUC, CGG, GCC, right? And then now we have the transcribed messenger RNA, and then each 3 letters is gonna be a codon, and then it's gonna code for anticodon, so the A becomes U in RNA, U is A, C is G, and then we have UAG, and then the tRNA, it's gonna bring the complementary amino acid for UAG, which is isoleucine, and then that's 1 amino acid. And then GCC is CGG for the codon, the anticodon is CGG, it codes for alanine amino acid. Now that's a 2nd amino acid, it's gonna come, hold hands with isoleucine, and then we're gonna have this polypeptide chain of amino acid until we hit the stop codon here. When we hit the stop codon, then the amino acids are gonna be released and they're gonna go take the shape of a protein. Okay, so what we're seeing here is, this is the real image of how it happens. So everyone, do you see the ribosomes? We have the small subunit, the large subunit, and then do you see the messenger RNA? And then it's starting with AUG. AUG, is the start codon, right? It codes for the methionine amino acid. So we have the E, P, and A. So basically, it comes here to the A, and then it goes down to the P, and then you have the amino acid. Then here, when it reaches to the E, the tRNA comes to the E, and then it exits from the E because we recycle those tRNA. It goes and grabs another amino acid. It goes and grabs another amino acid. And then another tRNA comes, the one on A, it shifts to the P, the one on P, it shifts to the E, and so on until we hit the stop codon, okay? So look, we start here, and then we continue. More tRNAs are coming with their anticodon and complementary amino acids. And then here we're making polypeptide chain. This is where the tRNA leaves to go get more amino acids. And then it keeps going until we hit the stop codon, which is the releasing factor. And then it releases the amino acids, right? And then we have, that's a primary structure of a protein. It's just made up of amino acid, but eventually it's gonna take the structure of a protein, okay? Yeah, so this is transcription and translation. Everyone, any question about transcription or translation? Do you think you understand it very well? Yes? Okay.
So why is UAG the stop code?
It's just that way. It's not only UAG, it's yeah, so I don't know how how they found out and decided these amino acids and then the table. As long as I've known this table is given, we're not even asked to memorize it. So there's UAG for a stop codon, there's UAA for a stop codon, and UGA for a stop codon. And then I'm glad that we have, you know, 3, 4 stop codons. The U's are the same, you know, the first letter is U. I don't know, like, you can still have like, like UAA or UGA, and then it will still code for the stop codon, you know. But let's say if it's UGG, then it's not gonna code for stop codon, right? And then if you don't hit the stop codon, you don't get the release factor, translation doesn't stop, and then you end up making the wrong structure of protein, yeah. But actually, I don't know how they came up with this table, but that's how it is. Yeah. Yeah, and then only AUG for start code, for the start codon is only AUG. Yeah. Thank you. Any other questions about transcription, translation? Okay, so since we're all comfortable with this information, this is a summary of everything we talked about. Who wants to like summarize this for, I'm joking, but this is what everyone, this is a big image of what we talked about, okay? So you can see everyone, like this is we're inside of the cell. Up there, that big circle is the nucleus. Do you see how the DNA transcription is starting, right? The introns, the exons, we're getting rid of the introns, the exons, the coding regions are getting together. We have the messenger RNA, and then do you all see the messenger RNA leaving the nucleus from the nuclear pore, right? And then the messenger RNA is gonna leave and go to the, where is it going? To the ribosomes, where what happens on the ribosomes? Translation happens. And then we see how we have the messenger RNA, the codons, the anticodons, complementary amino acids, they're coming together and they're making a polypeptide chains of amino acids where, and then, by the way, this is what a protein looks like, but if we break it down, it gets broken down into amino acid. I'm not gonna go into details here, it's not important, but amino acids, we have primary structure, secondary, tertiary, and quaternary. This is the primary structure. Secondary structure, they come together. We have the alpha or beta helices or beta pleated sheets, right? So basically they come together and then eventually they make this structure, okay? And then this is a protein. For example, all enzymes are made up of proteins. A lot of molecules are made up of proteins. And then the structure of the protein is very important because if it has the wrong structure, it won't be able to do its function, okay? And then once this protein is made, it's gonna go to its destination to serve its purpose, okay? But do we all understand the DNA, the genetic code in the DNA, the nucleus, transcription, translation, and how our genes are expressed? Okay, good. Next, we're gonna talk about the cell cycle, all right? So the cell cycle, everyone, is basically, okay. Here we can see the cell cycle. Reason why we're talking about this now, we've skipped to next topic. We've jumped to the next topic, which is mitosis and meiosis. We're gonna talk about cell division. I'm pretty sure all of you are familiar with it, but again, we're gonna go over it together, okay? So before we start mitosis or meiosis, right? So during the cell cycle, the cell spends most of its time in, what is this yellow part? In inner phase, okay? The inner phase is the largest phase of the cell cycle, okay? The cell spends more time in interphase than prophase, metaphase, anaphase, telophase, than in mitosis, okay? And this whole, like probably 80%, 90% is interphase. And then do you see where it says M phase, cell division, the orange part? That's where we have prophase, metaphase, anaphase, telophase, and then also cytokinesis. Remember, we have the phases, and then cytokinesis is when the cell, it divides into two, right? That's cytokinesis. So, but most of the time, the cell spends its time in interphase getting ready for mitosis. So everyone, look what happens in the interphase. Interphase is made up of three phases. What are the three phases that we're seeing here? We have the G1 phase, we have the S phase, and then we have the G2 phase, okay? So G1 phase is the cell starts growing, you know, so that it starts preparing for mitosis. Then we have the S phase, okay? The S stands for synthesis, DNA synthesis. What is DNA synthesis? This is where DNA replication is happening. And then on the next slide, we're gonna see what is DNA replication. And all of you do know what DNA replication is, but we'll go over it. So do you see up there, we have a DNA double helix, right? And then we're making exact copy of the same, okay? So basically, we're building the sister chromatid, okay? So we have one, right? And then we use this template to make another one, okay? And then they're both gonna be identical. And then we'll see it in the next picture. And then these are the two sister chromatids that come together. And then we have the X shape, which is the chromosome, which is made up of two sister chromatids, okay? Then we have the G2 phase. In G2 phase, it further grows more and it prepares for cell division. Now that it's grown, it's ready. Now we can start mitosis, okay? And then everyone, the phases of mitosis, the way that you can remember is the letters PMET, okay? P stands for prophase, M stands for metaphase, A is anaphase, T is telophase, okay? So PMET, that's how you can remember it. And then it's part of the mitosis. Interphase is part of the, well, the whole thing is part of the cell cycle, but the majority of the time we have the interphase, then we have the PMET, and then we have cytokinesis, okay? So this is the life cycle. Next, on the next slide, I'm gonna show you what happens here in the S phase. How do we make a replicated DNA? And then after that, we're gonna go over the phases of mitosis and cytokinesis, okay? So here in the next slide, everyone, what you're seeing is to make another copy of the DNA, we have to open up the double helix, right? And then use one of the strands so that we can make DNA to DNA. We can write down the complementary base pair and make another strand, okay? So to do that, of course, we need an enzyme, the helicase enzyme, which we call it the scissor, okay? It unwinds the DNA double helix. Do you see in the first picture how the double helix is getting unwound by the helicase enzyme, right? So it's opening it up. And then as it's opening it up, and then of course we have, just like in transcription, we needed RNA polymerase. Here in replication, we need DNA polymerase. It's an enzyme. A lot of the terms that ends with A's, it means it's an enzyme, okay? DNA polymerase. So basically, the DNA double helix unwinds. And then now that this is open, everyone, we have this DNA template, right? And then we have the DNA template over there. But the top and the bottom one, they have the same information because they're complementary base pairs, right? So basically, whenever we're gonna replicate a DNA, we're gonna use this strand and then this strand as a template. And then do you see how we're making a complementary base pair and building, you know, another one here and then another one here, where eventually this will become a DNA double helix, and then this will become DNA double helix, but they are identical because they're both made up from the same DNA base pairs, okay? Because those were like this line, do you see the T, right? That was, where is it? Yeah. Let's not do that, but what I'm trying to say is this strand here is complementary to this strand, and then we're making the strand complementary to this strand, and then we're making a strand complementary to this strand, okay? So now these two are identical, and then we ended up making one sister chromatid and two sister chromatid, okay? That's how our DNA gets replicated. So that's what's happening here in S phase. When you have DNA replication, it means we open one DNA double helix and then use the templates to make one here and then another one here, and then we end up with two. Okay, did you all see that? You see how we're opening it up? We're using the top and the bottom templates and then to add complementary base pair. But here we're doing DNA to DNA, okay? It has nothing to do with RNA. We're not doing transcription translation. This is just for cell division purposes, okay? This is how in the interphase, the cell is getting ready for mitosis, okay? And then we do need two sister chromatids, right? Because eventually after metaphase, during anaphase, the sister chromatids are gonna separate, right? And then they're gonna end up being in two cells, right? But they're identical because they were sister chromatids, because we replicated them here in interphase, okay? So here, everyone, that was interphase. Then we'll enter mitosis. So what is the first phase of mitosis? What's the first, what's the P from PMET? Prophase. Prophase is when the cell inside the cell, now it's ready to start mitosis. Prophase is the first phase of mitosis. Everyone in interphase, you'll see that the DNA is in threads, is in chromatin, but in prophase, you'll see that the chromosomes, they're gonna start condensing, the nuclear envelope is gonna start disintegrating. Do you see that happening in prophase? Because the chromosomes, they need to be let out of the nucleus so that cell division happens, right? We let the chromosomes out, we line them up, we split them up, and then we cut it into two. Now we end up having two identical daughter cells, which is the goal of mitosis, right? So, and then look how the centrosomes, they're traveling on opposite sides. Remember the centrosomes, centrioles, right? And they have started building this mitotic spindle or the spindle fibers. And then everyone, you can see here on opposite ends. In metaphase, now that these spindle fibers are built, you can see the chromosomes, they're lining up on the equator, correct?
Mm-hmm.
So that's what happens in metaphase. And then everyone, before we continue, we use mitosis so that for all the cells except for the sex cells, okay? We use mitosis to make new cells, or we use mitosis so that if we have a damaged cell and then we have to repair it, right? So for organisms to grow or repair, right, we need to replicate it, and then to replicate our cells, we go through cell division, which is these phases, okay? And then let's continue. So in metaphase, they line up on the equator, and then after that, we have metaphase, anaphase. What happens in anaphase? Yeah, so who separates? The sister chromatids, okay? So remember, the chromosomes are lined up, or let me do like this. The chromosomes are lined up on the equator, then the sister chromatids, they're going to separate, right? That happens in anaphase. And then are the sister chromatids identical? Do they have the same genetic information? Yes. And then after that, we have telophase. Everyone in telophase, now that sister chromatids, like we have the same genetic information on this side, we have the same genetic information on that side for the sister chromatids. Now a new nuclear envelope starts building up here, and then another nucleus starts building up there, and then the cell elongates more and more, because what happens in cytokinesis? The cell divides into two. Now we end up having two daughter cells, okay? So everyone, I want you to, take notes on what happens in mitosis. You can still see what I wrote this morning. In mitosis, we end up making how many identical daughter cells? Two identical haploid or diploid? Diploid, and then I'll explain to you what it is. Diploid daughter cells, okay? So everyone, what is 2n? What is 1n? N is the number of chromosomes, which is, n is a number of chromosomes, which is we have 23 chromosomes, right? And then, so n equals 23, okay? And whenever we have 2n, okay, which is diploid, okay? 2n means 2 times 23, it gives us what? 46 chromosomes. And then haploid is the opposite. Haploid is 1n, so it's 1 times 23, it's 23 chromosomes, okay? And then shortly, I'll tell you where do we need the haploid and where do we need the diploid, okay? But just remember, for mitosis, okay, is for cell growth, repair, right? How does an organism grow from like being a baby all the way to an adult organism? Mitosis, right? Our skin cells make more skin cells, our muscle cells make more muscle cells, our nerve cells make more nerve cells, right? And all of this happens through cell division, okay? Cell division is for all of our cells except for our sex cells, okay? Meiosis is strictly for the production of gametes, the sex cells, which is the production of sperm and egg, okay? And basically in mitosis, what we're doing is we're taking one cell, we're dividing it and making two identical cells, okay? And then in mitosis, cells are identical, right? Because let's say if your body needs to make more skin cells, all of your skin cells are gonna look identical, right? Or more muscle cells, all of your muscle cells are gonna look identical, right? But when it comes to sex cells, which is the gametes, sperm and egg, we're gonna talk about that shortly, okay? So also it's important to mention that in mitosis, we end up making diploid daughter cells, which means that they have 2n, right, which is 46 chromosomes, okay? Whereas in meiosis, we don't make sperm that has 46 chromosomes and then eggs that has 46 chromosomes. Why? Because during fertilization, right, you have the sperm and egg, they come together to make a new organism, the zygote, the first cell, and then that individual gets half of the genetic information from one parent and half of the genetic information from the other parent through sperm and the egg, right? So the sperm is going to give half of its genetic information. The egg is going to give the other half of its genetic information. So 23 chromosomes, the individual will end up having 46 chromosomes. But if the sperm was 46 chromosomes, the egg was 46 chromosomes, then the individual has 92 chromosomes, right? It's not normal, okay? That's how you can remember. In meiosis, where we make sex cells, which is the gametes for the production of sperm and cell, it only has half the genetic information, okay? And then that's why each parent gives half of their genetic information. Like you get chromosome 1 from one parent, chromosome 1 from the other parent, right? That's why you inherit both traits from both parents, okay? So I started talking about meiosis, but we're still here. So everyone, this is interphase, prophase, metaphase. They're lining up in the middle. So This lab, I am going into a lot of details, but remember the lab exam questions are mostly like labeling types of question. And then what we're seeing here is those are types of questions that you'll see. For example, I would give you an image of metaphase where all the chromosomes are lined up in the middle, and I'm asking you, oh, which phase is this, right? And then you would say metaphase. Anaphase, the sister chromatids are separating, and then telophase, you'll see how we have a nucleus forming around the sister chromatids, and then we have cytokinesis where the cell divides into two, and we end up making two identical diploid daughter cells. And then this is a summary of everything that we talked about that takes place in mitosis. Now in meiosis, actually, there's a video I want to share, but I'll share mitosis and meiosis together. In meiosis, everyone, as you know, humans, they reproduce through sexual reproduction, which mixes the DNA of the egg and the sperm together. And then in order for this to happen, we get half of the genetic information from the egg, and then we get half of the genetic information from the sperm, right? And then for this to happen, the body needs to make sperm and egg, right? So that so that all of this can happen, right? And then the process responsible for the production of sperm and egg is gonna be meiosis. Whenever we make meiosis, and then I'm gonna write on the board, in meiosis, we end up making 4 non-identical haploid daughter cells, and then which eventually they're gonna turn into sperm and eggs, okay? It's strictly for making sperm and egg are sex cells. And then meiosis is similar to meiosis, is that when cells enter meiosis, they go through interphase, they go through G1, S, G2, and then they enter meiosis, okay? And it also goes to prophase, metaphase, anaphase, but the only difference is in meiosis, we have meiosis 1 and we have meiosis 2, right? And then we're gonna see what the difference is in meiosis 1 and meiosis 2. And besides that, something happens in meiosis that it does not happen in mitosis, which I want to talk about next, okay? So I want everyone to think about this. Have you noticed that, I don't know if you have biological siblings or if you know a friend or anyone that has a biological siblings, it means that they share the same parents, right? And I'm not talking about identical twins, right? Isn't it strange when you think about it that two individuals, right, they have the same parents? It means that they got half their genetic information from one parent and the other half from the other parent. But these two individuals, they don't look identical, but they both inherited, half the genetic information from the one parent and half of the genetic information from the same parent, right? So they inherited both parents half of their genetic information from the exact same parents, but they don't look the same, right? For example, if you do have biological siblings, do you look the same? Not talking about identical twins. Or is there anyone on this world that looks identical, in this world that looks identical to you? No, each human being is unique, and then there's no one that looks like you, right? So something is happening, right, that is creating this genetic diversity, right? Who can tell me what happens in meiosis? And then you probably know this. So if you think about it, you'll remember what happens in meiosis that does not happen in mitosis that creates this genetic variation. What happens in meiosis? I think when I say it, you're gonna know what it is, but you haven't thought about it this way. Okay. How many of you are familiar with crossing over? Crossing over is when genetic information gets swapped, okay? So something happens in meiosis that doesn't happen in mitosis is crossing over. So basically, do you remember how I said, like, we get half of the genetic information from the sperm, the other half from the egg, right? For example, the sperm gives, by the way, can I erase this? Are we okay with the DNA and what it looks like? Okay. So let's say, okay, this is enough. So let's say, the egg gives a chromosome 1, and then the sperm gives, chromosome 1, and then during, prophase, what happens is the homologous chromosomes, they pair up, they try to find each other, like chromosome 1 from one parent finds the chromosome 1 from the other parent, and chromosome 2. So this is how we get the homologous chromosomes in meiosis. But something called, crossing over happens where everyone, do you remember we have our DNA, and then here on our DNA, remember, this is a DNA double helix, this is DNA double helix, it has all these letters on it, right? It has all the genes, right? So, sometimes whenever these homologous chromosomes, they pair up, something called crossing over happens where, portion of the DNA gets swapped. So, this part, right, gets swapped with this part. Okay, so like this. Do you see that, right? And then remember, this is a sister chromatid, and then this is a sister chromatid, right? And then this is a sister chromatid, and then this is a sister chromatid, right? So, now, and then remember, in meiosis 1, okay, we get this, crossing over, this swap, right? In prophase 1, and then in metaphase 1, these chromosomes, these homologous chromosomes, they line up in the middle, right? During metaphase. And then during anaphase, these chromosomes separate, okay? So whenever we're separating, it means that one side we're getting, this one, and this one is going on one side, right? And then to the other side, we're getting, oh my God, this is hard to draw. We're getting, the other side, right? So in anaphase, the sister chromatids don't separate, the homologous chromosomes separate for meiosis 1, okay? And then they separate, and then, eventually we end up having, two cells, and then now in meiosis 2, now, these chromosomes are gonna line up in the middle, and then the sister chromatids are gonna separate, right? So if the sister chromatids separate, what is this? This is all green, right? This is, green and blue, right? This is blue and green, and then this one is all blue, right? This is when the sister chromatids, they separate, and then each one becomes a cell, and then these cells are gonna go further and become sperm and egg. Okay, so you can see here in meiosis 1, prophase 1, the homologous chromosomes come together, crossing over happens. Metaphase 2, the homologous chromosomes line up in the middle, and then the homologous chromosomes, they separate, but you can already see the crossing over between the two colors, and then it goes through telophase and cytokinesis. Then each one goes through meiosis 2. Meiosis 2 is very similar to mitosis. Look, the chromosomes line up in the middle in metaphase 2. The sister chromatids, they separate here and here, and then each one becomes a cell, right? And then, but they are identical or non-identical? Are they identical?
No.
They're not identical. Okay, so that's why in biological siblings, you see that, like, for example, if there are 4, you'll see like 2 of them, they're like exact opposites. Like one is all blue, one is all green, right? They have nothing to do with each other. But we also see siblings that, look, for example, this one has this green matches with this green, right? So like these 2 siblings, like, oh, they have some similarities, but some things are different, right? Or this person has some similarities with this person, or this person has some similarities with this person, right? So that's why between, biological siblings, you do share, some resemblance, and then sometimes we say that, oh, they're completely opposite, right? So, even though we share, half of genetic information from each of the same parents, from the same biological parents, but because of crossing over, right, we have this genetic variation and we don't look identical, even though we share the same parents. This is a review on the summary for meiosis phases. And then everyone, this slide is explaining what is crossing over. Basically, when the chromosomes, parts of the chromosomes get swapped, and then with those chromosomes, those genes, the sequences, right, they also get swapped. Sorry. This happens in prophase 1, meiosis 1. Okay? And then this is the continuation of meiosis 2. Okay, we only need like 2 minutes or 5 minutes for the next section. But before I give you a quick break, I want to play 2 videos for you. And then I think after, it's in our additional resources in lab. I think you'll feel really good about mitosis, meiosis after you've watched this, okay? So if you go to lab 2, so the handouts I gave you, they're here. The slide that I'm sharing, it's here. If you go to lab 2, additional resources, this first video is good. It's on transcription translation. This is on meiosis. This is video on how to use a microscope, which is the last thing that's left for us to talk about today. And then I'm going to play these 2 short videos. They're very short, to the point, very old, but they're very good, okay? That's why I keep playing it. So, yeah. It takes a minute for the sound. Any minute now.
Having already replicated. Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes.
Look how chromatin, from chromatin, it went to chromosomes.
Protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles.
What is this phase? Anaphase, right? The sister chromatids are separated.
Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform.
What is this?
With mitosis complete, the nuclei and cytoplasm separate, creating 2 new cells in this ongoing cycle that sustains all life.
So when it separates, that is what? Cytokinesis. Good. All right, and then this one.
Taxes was waiting and wondering.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The 2 homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart.
By the way, we started meiosis 2, right? This is anaphase 2.
This leaves the sister chromatids intact. With
No, it's still one. Sorry, it's still one.
twice the amount of genetic information the cells will need, nature must cut the genetic deck in two.
Okay, now we're starting meiosis 2. This is prophase, and then prophase 2, and then metaphase 2. We'll see when the chromosomes line up in the middle. Sorry, I zoned out for a second. This music is putting me to sleep. I feel like I'm being hypnotized.
The second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
Okay. All right. So that's the additional information. Everyone, we only have like a couple of slides left on how to use the microscope, and I don't need a lot of time for that. I want to give you, it's 4:10, like just 5 minutes break. Probably if we do that, we'll finish the class earlier. Let's take a 5-minute break. Like if you need to go to the restroom, like, and then I'm saying 5, it turns into 7. So until we get started. If you look at the handout, question 1 and question 2 in the handout, one is looking at letter E under the microscope, and one is putting the ruler under the microscope. Those are, I want you to do it hands-on in lab. Okay, so go ahead and take a 5-minute break. Or if you don't want to take a break, but if you want to walk around, go ahead and grab a microscope from the cabinets. And then when we get started, we're just going to talk about, it's only a couple of slides on microscopy, and you've seen this in Bio 5, okay? So yeah, take a couple of minutes and then we'll start shortly. All right. All right, everyone, we're gonna talk about how to use a microscope. So all of you in front of you, you have a microscope. So feel free to like look around that microscope while we talk about the microscope, okay? So first, I should have probably shown this slide first. This is how you hold and transport a microscope. You have to hold it with 2 hands. I wasn't paying attention. Did you hold with 2 hands? One, you hold the arm with 1 hand and you support the base with the other one. Okay, so that's how you transport a microscope. Also, before you start using your microscope, tell me if the previous students, they stored the microscope in the right way. Is the stage lowered to its lowest? And is the objective lens on the 4 times the lowest objective lens? Okay, so whenever we're gonna put the microscope back, we have to make sure that the stage is at its lowest. So you can play with the coarse adjustment knob, the big knob, to bring it all the way to the bottom, right? And then also the objective lenses, it has to be on the 4 times, okay? Which is the scanning objective lens, okay? So whenever you're gonna put your microscope back, make sure that the stage is low as it is at its lowest, and then the objective lens is on 4 times, and then the plug organized, and then you'll put it back where you got it from. Also, another thing, don't forget slides on the microscope, okay? Return the slides back. Everyone, this is a compound light microscope, the one that you're seeing in front of you. The reason why it's called compound means more than one, because it has more than one magnifying lenses, okay? One is the ocular lens, the one that we look at it through our eyes. Everyone look at the ocular lens, the one where you look through your eyes. Do you see a number, magnification number? Do you see the number 10 times when you look at the ocular lens? Everyone look at your microscopes. Do you see it's written 10 times where you have the eyepiece? Where you have the ocular lens, right? Do you see 10 times? Do you see that number? Okay, so compound light microscope has a magnification 1 at the eyepiece, which is the ocular lens. And then on top of that, it magnifies further because we have the objective lenses. Do you see the 4 objective lenses, right? Let's read the the numbers on the 4 objective lenses in case you can't remember it from Bio 5. So what's the smallest one? 4 times, right? It's called the scanning lens. And then what's after the 4 times? 10 times. That's called the low power objective lens. And then after the 10 times, what do we have? 40. 40 is the high power objective lens. And then the last one is 100 times. That's the oil immersion. We're very careful when we're gonna use the oil immersion. We don't put the oil immersion lens unless we have oil on the slide. And then we slowly bring the oil immersion lens because that's like the longest one. Do you see it? It's the biggest one. So it can hit the slide and it can break the slide or the lens can be broken, okay? So we need a drop of oil and then we slip it and then we look at it under the microscope, okay? And then today we're not gonna use the 100, so we don't have oil. Try to stay away from the 100, okay? So we have the objective lenses, everyone. We have so this is the head, this is the arm of the microscope, objective lenses. This is the nose piece, that's where you turn to see the different objective lenses. This is the mechanical stage, so you can control the stage going up and down with the coarse adjustment knob. And on the side, you have these stage control where you can control if you want to go left and right, up and down. Everyone, this is where the light source comes out. This is the condenser. We can control how much light do we need it to pass. This is the light switch, brightness, and then that's the base, okay? And then, so because it's a compound light microscope, we said that it has 2 sources of magnification. One is the ocular lens and one is the objective lens. So if I ask you what is the total magnification, what you do is you multiply the ocular lens by the objective lens, okay? So what did we say? What's the magnification of the ocular lens? 10 times, right? So as long as we're using this microscope, it's gonna be 10. And then if I'm using the scanning objective lens, the shortest one, that is 10 times 4. What's my total magnification? 40 times. If I'm using 10 for the ocular and I'm using the low power, which is the 10 times 10 is 100, 100 is the total. If I'm using high power, which is the 40, I'm looking at it through the 10 times ocular lens. 10 times 40 is 10 times 40 is 400. And then if I'm using the oil immersion, which is 100 times, and I'm looking at it through the 10 times ocular lens, 10 times 100 is 1000, okay? Good. And then how do you use the microscope? The microscope should have an on and off switch. It should have a plug. We have to plug it in. This is the ocular lens and the 4 objective lenses that we talked about. We have the coarse adjustment knob and the fine adjustment knob. Everyone, the coarse adjustment knob is the bigger one. And then I think our microscopes, they look a little bit different than the picture one. You have your coarse adjustment knob, it's the bigger one, and then more closer to you, you have the fine adjustment knob. It's the smaller one. Do you all see your coarse and fine adjustment knobs, right? The bigger one is going to be the coarse adjustment knob, and then that smaller one is going to be the fine adjustment knob, okay? If you play with the coarse adjustment knob, you'll see that your stage is going up and down, okay? And also you have your microscope stage, and then on the stage, you have these clips where you insert your slides. A lot of times I see students putting the slide wrong. Make sure that you place it right. If you click here, this will open up and then the slide should sit here, exactly this shape, okay? If you open this up, that's where the slide should sit there, slide in and have the right shape. Yeah, so, and then also here, this is a condenser. Basically whatever the light is, so when the light comes through and then under the stage you have a condenser, you can control dark field, bright field microscopy, or how much light you want it to pass by. And then whenever we're using a microscope, you'll put your slide, and then we're gonna start with the 4 times. You don't start with 100 times, 40 times, 10 times. You start from the smallest one, and then you use your coarse adjustment knob, and then you turn it until the slide goes all the way up, right? And then that's when you'll start seeing something like what you're looking at, but it's not gonna be super clear. So you use the fine adjustment knob. You use the fine adjustment knob to make it clear. And then only once you see it clearly, clear, good image, then you go from 4 times to the next objective lens, which is what? 10 times, okay? When you go to 10 times, you might or might not use the coarse adjustment knob. So if you're seeing it, then good, don't use the coarse adjustment knob. If you're seeing it and it's blurry, use the fine adjustment knob so that you can see it clearly, okay? After you have a clear image at 10 times, then you go to 40 times. If you don't have it clear, then you don't go up. After you go to 40 times, at 40 times, we do not use the coarse adjustment knob, okay? Because once you've hit 40 times, it means you're magnifying 400 times in total magnification. So if you move it, then you're gonna lose what you're looking at, okay? So at 40 times, you don't use the coarse adjustment knob. You only use the fine adjustment knob to see it clearly. And then after that, if you're gonna use the oil immersion, which we're not gonna use it today, before going to the oil immersion, between 40 and 100, you put a drop of oil, and then you slowly slip the oil immersion, and then it's not gonna break anything because there's the oil there. And then when you put the oil, do you use the coarse adjustment knob? No, right? We only use it at 4 times and maybe 10 times. We don't use it at 40 times, and we absolutely do not use it at 100 times. We only use the fine adjustment knob because by accident, if you use the coarse adjustment knob and you lost what you were looking at, right, you have to either start all over. If it's a ready-made slide, you have to wash the slide, clean the slide, and start over from 4 times. Or if it's a slide that you prepared, then you have to make a new slide. And then I'm not talking about, for example, let's say in microbiology, you're gonna make your own slide, right, smear, and then it takes at least 10, 15 minutes to put the bacteria on the slide and fix it and stain it and be able to see it under the microscope. So after all that work, you don't wanna mess it up, okay? So once you put oil, you don't put, you don't use the coarse adjustment knob, only the fine adjustment knob, okay? Yeah, so you start with the 4 times and all that. And then this is how you're seeing the real image under the microscope. And then, yeah, so the fine adjustment knob every time, everyone, sometimes we use it because sometimes you have like different layers, right? And then to be able to see the different layers, you can play with the fine adjustment knob. Another important information here is in microscopy, the more you increase the magnification, like the more you go from 4 times, 10 times, 40 times, your field of view decreases, okay? So, and then think of it this way. Let's say we all take a group photo, right? And then you take the phone and then you magnify it on yourself to see how you turned out in the picture, right? But when you're magnifying it on yourself, do you still see the whole group in the picture, or you're decreasing your field of view, but you're magnifying what you want to see? You're decreasing your field of view, okay? The microscope works that way. For example, for the first question, I believe it asks us to look for letter E under the microscope at 4 times, 10 times, and 40 times. At 4 times, we're gonna see the whole letter E. At 10 times, we're gonna see lines that looks like letter E, right? And then at 40 times, we're probably just gonna see like a black line, which we're not even gonna tell which part of letter E it is because of how magnified it is, right? We don't even know like where it magnified it all the way. So as you increase the magnification, the field of view decreases. And then another thing is, we're not gonna do this today, preparing a wet mount. The third question asks you to swab your cheek cells and then stain it and then look at it under the microscope and draw the picture. I'm not going to ask you to swab your cheek cells, but for question number 3, you can look it up on the internet, like a slide under the microscope, or micrograph, which means a picture of a microscope slide, a micrograph of cheek cells, and then whatever magnification that it is saying, and then you can draw that, or you can upload that image as your answer for question number 3, okay? But question 1 and 2, we're gonna do it together in class. Does anybody have any questions about microscopy before we start working on the letter E and the ruler? All right, okay, so